In the realm of modern medicine, chemotherapy stands as a formidable weapon against cancer, but its potent effects often come at a cost. Chemotherapy, while targeting rapidly dividing cancer cells, can also harm healthy cells, particularly those responsible for producing white blood cells, the body's primary defense against infection. This collateral damage can lead to a condition known as neutropenia, leaving patients vulnerable to potentially life-threatening infections. Enter Neupogen, a medication that has revolutionized cancer care by mitigating this risk and improving the lives of countless patients undergoing chemotherapy. Approved by the FDA in 1991, Neupogen, also known by its generic name filgrastim, is a synthetic version of a naturally occurring protein called granulocyte colony stimulating factor, or GCSF. This remarkable protein plays a crucial role in stimulating the bone marrow, the body's blood cell factory, to produce more infection-fighting white blood cells, specifically neutrophils. The development of Neupogen marked a significant advancement in cancer treatment, offering a way to bolster the immune systems of patients weakened by chemotherapy. By increasing the production of neutrophils, Neupogen helps to prevent febrile neutropenia, a serious complication characterized by fever and a dangerously low neutrophil count. Prior to the advent of Neupogen, managing chemotherapy-induced neutropenia often involved prolonged hospitalizations, intravenous antibiotics, and dose reductions or delays in chemotherapy, potentially compromising treatment outcomes. Neupogen emerged as a beacon of hope, reducing the incidence and severity of these complications and allowing patients to tolerate higher doses of chemotherapy, potentially improving their chances of survival. The production of Neupogen is a testament to the power of biotechnology, specifically recombinant DNA technology. This groundbreaking technique allows scientists to insert specific genes into the DNA of living organisms, transforming them into tiny factories that produce valuable proteins like GCSF. In the case of Neupogen, the gene responsible for producing human GCSF is inserted into the DNA of the bacterium Escherichia coli, or E. coli, a common and easily grown microorganism. The genetically modified E. coli are then cultured in large fermentation tanks, where they diligently churn out copious amounts of GCSF. This process ensures a consistent and reliable supply of this life-saving protein, free from the limitations and potential impurities associated with traditional methods of protein purification from animal sources. Recombinant DNA technology has revolutionized the pharmaceutical industry, enabling the production of a wide range of therapeutic proteins, including insulin, growth hormone, and clotting factors, to treat a variety of diseases. Neupogen stands as a prime example of the immense potential of this technology to improve human health. Neupogen has emerged as a cornerstone in the management of chemotherapy-induced neutropenia, providing significant benefits to cancer patients undergoing this aggressive form of treatment. By stimulating the production of neutrophils, the body's first line of defense against bacterial and fungal infections, Neupogen significantly reduces the risk of febrile neutropenia, a serious complication that can lead to sepsis, a life-threatening condition characterized by a systemic inflammatory response to infection. Studies have consistently demonstrated that Neupogen significantly reduces the incidence, duration, and severity of febrile neutropenia in patients receiving chemotherapy. This translates into fewer hospitalizations, shorter hospital stays, and a reduced need for intravenous antibiotics, ultimately improving patients' quality of life and allowing them to focus on their recovery. Moreover, by mitigating the risk of infection, Neupogen enables patients to tolerate higher doses of chemotherapy and complete their prescribed treatment cycles on schedule. This is particularly important in cancers that are highly responsive to chemotherapy, as maintaining the intended dose intensity and treatment duration can significantly impact treatment outcomes and long-term survival. While the benefits of Neupogen in supporting cancer patients during chemotherapy are undeniable, the cost of this life-saving medication has been a subject of ongoing debate and concern. Neupogen is a biologic drug, meaning it is produced using living organisms and complex manufacturing processes, which contribute to its high price tag. The cost of a single Neupogen injection can vary depending on the dosage and individual patient factors, but it is not uncommon for a course of treatment to amount to thousands of dollars, placing a significant financial burden on patients and healthcare systems. The high cost of Neupogen has raised concerns about access and affordability, particularly for patients without adequate insurance coverage or those facing financial hardship. Some patients may have to make difficult choices between their health and their finances, potentially compromising their treatment and overall well-being. Efforts are underway to address the issue of Neupogen's cost, including the development of biosimilar medications. Biosimilars are highly similar versions of biologic drugs like Neupogen that are produced after the original drug's patent has expired. Biosimilars undergo rigorous testing to ensure they are as safe and effective as the original drug, but are typically available at a lower cost.